Hey Rajesh,
Hi, good morning.
good morning. How are you doing?
Doing well. Yeah.
Okay,
How
okay.
do I sound?
You sound good.
Okay. Yeah.
That's strong,
Hey, I'm
yeah.
recovering.
Hmm. Is Sweekriti there or she left?
I think she was there.
I see. Not sure. Yeah. Anyways, that's fine. That's good. I went to the next link. I mean, link for the next class.
Next class. Okay.
Nobody is here. Yeah. Good. That's good picture. Looks like
Yeah,
a
I
lot
found
of.
it on Google. Whenever you Google for stem cells, you get something very exciting.
I see.
Yeah.
Yeah, they look. I thought they were ice crystals or something.
Yes, they do give that impression for sure.
That's interesting. Is that the chromosomes inside?
Yeah, I think they are trying to picture a, a good nucleus, and I think the coloring is just to impart that
Hmm.
there lots of things are sort of fluid when it comes to stem cells, and they
Hmm.
can take up a fate, and having no shape and form in sense that they look sort of globular,
Hmm.
uh, based on the picture that given with an opportunity they can become whatever they want to based
Hmm.
on the local signaling. Yeah.
Yeah, yeah, yeah. I think that's a good point. Actually, it's possible that the nucleus
Yona,
is. Oh, yeah.
yes, for the Saturday thing, you gave some link, no, for the, I think, presentations.
Mm-hmm, mm-hmm.
Actually, I have exams, so Saturday is not right for me.
I see. I see.
Even I
So,
have exams.
okay, who is the second one?
Shumoy, Shumoy.
Okay, good, good. More? I think, I was going to respond to Manisha, but please let us know how many of you have exams on that day. So based on that, we can take a call.
And Nag, uh, the two, there are 4 people in my group for the presentation day, like in group number 2, but 2 out of those 4 I've never seen in my life in the lectures. Like, I don't know who they are. They
And in
don't
my
attend. I don't
group,
know.
I haven't seen many of them. Many of them.
It's only me and Halak that I know of. Other than me, there's only Halak that I've seen in the attendance. Because Shonika and some other, like Ankita, I've never seen them or talked to them or anything. So I don't
Oh,
know if
Ahana,
they'll even attend.
they are pretty regular. Ahana, they're almost
Not
every.
Ahana,
Uh,
Ankita and Shonika.
yeah, 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 yeah. Yeah. So we'll talk to them. So then, yeah, they're there.
So I don't even know if they'll come that day. So I don't know.
So that's something I think we should talk about. I guess after the class, I think we should not disturb Rajesh's.
Yes,
Yeah.
yes, of course.
Let's keep the
Excited for
excitement for the
Raj's
final.
lecture.
Yeah, absolutely. So we'll do that after the class. Yeah, yeah. Good, good. We'll discuss about it. Thank you for reminding all of you. Yeah. Yeah, coming back to the nuclear thing, Rajesh, I think it'll be
Yeah.
interesting to see. I'm not sure because, all the cells are not stem cells, but also, actively transcribing.
Yes.
They have a very interesting nuclear shape indentations of the nuclear membrane, and pores are majorly positioned towards that side. And it has
Yeah.
usually a lot of cytosol on that side. So
Yeah.
there is a particular orientation. I'm pretty
Yeah.
sure people have done a lot of research on that, but I'm not sure if they cared much about, if they did a lot of work on stem cells themselves.
Yeah. It
Yeah.
seems like when it comes to hematopoietic stem cells, the amount of nucleus, the area of the nucleus in general, the volume,
Hmm.
in comparison to cytoplasm is very different. There is hardly any cytoplasm when it comes to hematopoietic stem cells. On the contrary,
Hmm.
if you see megakaryocytes and other cells, they have huge nucleus and lots of cytoplasm.
Hmm.
And same is true for, if you consider B cells or T cells, they have
Hmm.
a good distribution of cytoplasm. So there are a
Hmm.
lot of things change once a
Yeah.
cell decides, okay, I want to be B cell or T cell or a red cell. Like in case of red cells, what we know, at least in case of humans, there is no
Hmm.
nucleus.
Hmm.
And all the protein which is there present in the cytoplasm is just hemoglobin.
Hmm.
And it's just terrific to see how what cell does to pumping so much amount of globin
Yeah.
and at the transcriptional level and then eventually translational. It seems there are specialized ribosomes
Hmm.
uh, which are made during erythropoiesis, which is red cell formation. So it's just amazing. Yeah.
Yeah. Yeah. Awesome. Yeah, I think we're almost into 10 minutes. I think we'll go ahead and start, I guess.
Yeah.
Before that, guys, I'll introduce you to Rajesh because most of you do not know him, right? So probably many of you seeing him for the first time. Good. That is a good thing, right? You already know him very well. Creates nice fluidity into the class. Very
Very
good.
exciting.
Very exciting, right? Yeah.
Yeah.
So I don't know, a lot of things you all know now that, oh guys, all of you are from NCBS, you know your friends and everything. Yeah. So that's there. But something with Rajesh and I are we're not classmates, sorry, we're lab mates in Vijay's lab. So that's where we both started around roughly at the same time. We were both refugees to Vijay. And we both started around some same time.
Yeah.
But things were going well, and I went to Israel on my way. By the time I come back, all that I knew was Rajesh found these mysterious cells that were always in our backyard, and we did not know about them. And they turned out to be stem cells. And by the time I came back, all that I knew was Rajesh's work got reviewed in eLife, got a fantastic reviews, overwhelming response, and yeah, and it was on the verge of publication. It was beautiful work, and how he discovered those and then figured out that there were stem cells. There was a very good response all over the institute actually in NCBS. And then after that, he went on to investigate whether they were there even in the adult muscles, because these were discovered during the larval life where adult muscles were made. The pupil adult muscles were made, but whether they were kept in muscles for regeneration purposes later in the adult life, actually, when the muscles are injured. So yeah, so then Rajesh went ahead and investigated, injured those muscles, and showed, I think for the first time, that there is these stem cells in Drosophila in muscles. And that was again another beautiful discovery, that to say that there are stem cells there, they after the injury they regenerate, they proliferate, and then they start to heal the muscle. So these back-to-back papers on stem cells in muscles, both during development as well as in adult life, I think it was not one of the common things that happens in every PhD. Um, it was great. It was a great discovery, both of them. And we were all very proud of that as a lab, and Vijay was very proud of it. And I think particularly at that time around, it was a good, I would say, good mix of people in Vijay's lab. It was a glory was back to Vijay's lab in some ways around that time. So because in the beginning, Vijay had this flurry of papers that described about muscle development in general, and then they were highly cited. But later there was a pause and there was a silent period, I would say, and then there was this again a good time in the lab that there were a lot of good papers on muscles came out around the same time. Yeah, so it was fantastic. And then we never thought that we both will end up back again in Harvard. So it's good. It's all good, and I'm very glad that Rajesh is part of that this course. And I'm sorry to keep you guys waiting for like I don't know, almost 2 months into the course, right, for this lecture. So, but when we start the development course, so there Rajesh will teach maybe I don't know second class or third class or something. So it depends on
Looking forward to that too.
yeah.
Awesome.
Okay, so with that, Rajesh, all yours.
Thank you.
Yeah, take it away. Yeah.
Yeah. So thank you, Nag, for that generous introduction. I definitely enjoyed our time together on campus, and those days were amazing. But today we'll be talking about a slightly more general aspect of stem cells. Now I'm currently postdoc at Children's Hospital in Lenzon's lab, and we'll be learning about stem cells and its tremendous potential in organ repair and during aging process. So here is a beautiful video of zebrafish embryo development. And as you can see, it begins as a single cell. And in matter of hours, this video is speeded up, but in the matter of hours, as you can see, it forms multiple number of cells. And these cells eventually will form adult zebrafish. The whole process in real life takes around 24 hours, but here you can see it happening in front of your eyes in no time. The GFP in this organism marks the vascular cells, and you can see there is a time during development when the GFP reporter is switched on and it marks the vascular cells. Now, this is the process. A lot of us think that it's one directional, which means from a single cell, this whole organism is formed. And the stem cells which were involved in this are sort of lost. And the real question is, can we actually take these fully differentiated adult cells and walk them back in terms of development counterclockwise? And can we really get the stem cells so that if there is a need for us in terms of tissue repair and organ damage, we can take those stem cells and put them back to restore the tissue function and organ function. So in other words, I'm going to play this another video which shows exactly opposite of what we just saw. It begins as a full zebrafish, fully developed, and it goes back to the single cell status. And now you can imagine the kind of processes involved in this from adult stages of the development to the single cell organism, which is a stem cell, which really gives rise to the whole process. And the real question is this process a reversible process? In that sense, can you take cell from one stage of development and really turn it around and take it to a previous stage of development? Which means, can you turn them back into the what they call the stem cells? Now, stem cells are defined as cells which are capable of both self-renew and ability to differentiate. There are a lot of work done by Morrison, Till, and McCulloch, and Wiseman, who are at different labs around the globe. Wiseman has a lab in Stanford. And according to them, stem cells actually provide a fantastic opportunity to investigate mechanisms that regulate embryonic development, cellular differentiation, and organ maintenance. Now, given with their ability to proliferate and form different kinds of cells, they have always been subject of intense research. And according to lots of scientists and my colleagues here at Harvard Medical School, we think that they can be used for novel cell-based therapies when it comes to aging and organ damage and repair. Now, the term stem cell actually originated in the two major contexts when it comes to biology. One of the major contexts was germline stem cells, which gives rise to the gametes. And this term was coined by Theodore Boveri and Valentina Haeckel. And they use this term to describe germline stem cells. The another context, as shown here on the slide, was hematopoietic stem cells. And this is the same time during which Arthur Pappenheim, Alexander Maximov, and Ernst Newman were also trying to understand how actually this stem cell, here shown at the center in this schematic, is actually able to give rise to these diverse kinds of cells as shown here in the schematic. And this is the time when scientists were thinking about stem cells and all sorts of possibilities related to stem cells. Now, when we say stem cells and where they are actually found, we find that stem cells are plenty in the nature. In every process that involves growth and regeneration actually involves some or other kind of stem cells. So here is a video of a forest which is coming out of its quiescence and growing. And you can appreciate that every move a plant makes involves these stem cells known as meristem stem cells. So here's a video for your consideration. Now, as we saw, actually every part of the plant, when it comes to the time of growth and change in season, has an ability to spring back to life. And when this was essentially, seen time after time, there's one scientist who was a Swiss botanist. His name was Carl Nageli. In 1858, actually coined the term meristem, and it is actually something we found out. It's a combination of meristos, which is a Greek word for an ability to divide, and stem, phloem, and xylem. The suffix for the stem comes from, literally using it for the cells which are actually able to divide. And if you see in terms of the meaning of these stem cells and meristem, it's very similar in terms of what their abilities in terms of divide and form new organism and new organ in general. And though these terms are actually corresponding to the same ability, their origin seems to be entirely different. So you can see people are making this observation during history and wondering what are these cells which are capable of giving rise to all sorts of cells in the body and in the plant kingdom. Now, the stem cell term actually appears repeatedly when it comes to the biological literature, and it was first coined by this great scientist whose name is Ernst Haeckel. And Ernst Haeckel was working on biology and around 1868. And this is the same time when lots of other bigger discoveries were made. And this was the same time when Darwin was making his discovery. So Ernst Haeckel was a big proponent of uh, Darwinism, and he was one of the biggest scientists when it comes to Germany. He was actually involved in drawing lots of these evolutionary trees, and those trees which connected one organism from cellular form to another slightly complicated form. He called those trees, which are very common when it comes to evolutionary studies, called them as the stem. And this really refers to the stem trees in if one has to translate that term from German to English. In the same context, when he was drawing those trees, the first organism at the base of the tree, he called that as a stem cell, which essentially literally means stem cell in German. Now, there is an interesting thing about Ernst Haeckel. Besides being a botanist, he was also a naturalist, biologist, philosopher, and physician. And he was a really impressive scientist. He was born in Germany around 1834. And you can imagine around those times you could graduate faster. So he graduated in 1857. And by 1860 or so, he was already running a lab. And during early his career, like yourself, he found amazing mentor. So his mentor, Johns Muller, took him to an expedition to see these marine organisms in the North Sea near Heligoland. And this was one of the profound experience he came across. And this is where his real interest for biology actually was something which started, and the lifelong fascination for natural forms and biology started. In 1859, he took a trip to Italy in Napoli, and that's where he actually refined his artistic skills. And this is the same time he went on to study these unicellular protozoans known as radiolarans, as shown here in this particular picture. And now you can see he was so good at drawing, he could draw the exoskeletons of these radiolarans, which are unicellular organisms, and they are very limited by their own experience about environment by these projections he drawn. And this was published. There are about 59 such illustrations. If you guys get time, do find those illustrations. They're very beautiful, very inspirational in some sense. And they were published in this book known as Kunstforming der Natur. It's available online, and I would say most of you will like it. During the same time, around 1859, 1860, he published a book which talked about a lot of evolution. And this book became sort of a handbook when it came to knowledge of the biology. And the book's name was Naturliche Schöpfungsgeschichte. And this is something people referred back again and again when it came to the First World War. And he was really the leading authority when it comes to biology. Now, this guy gets a lot of importance when it comes to stem cells because the term he has coined is STO. It is not only a historical relic, but it's also actively used by the stem cell scientists till today. Now, having said that, what we know from the research done till today and from biology textbook, that when it comes to early stages of development, as shown here, there are two gametes involved in formation of life. There is a oocyte, and then there is a sperm cell. And both of these cells actually are formed by the respective germ stem cells. Now, at every stage of development, when the fertilization happens, it leads to a series of active cell divisions. And at each cell division, the cells kind of lose their potential to form different kinds of cells. And this is referred to as development. And the cell which is actually formed right after the fertilization is actually totipotent, which is able to give rise to the whole organism. At the same time, when the cell processes and then passes through different stages of development, during the blastula stage, it forms this inner mass of cells known as embryonic stem cells. And this is something which forms a key when it comes to organism development. Now, at this point, embryonic stem cells forms a core of attention when it comes to stem cell discoveries and stem cell use when it comes to therapies. And what we know from the lot of research from Daily Lab and others, that embryonic stem cells are capable of giving rise to different kinds of cells, as shown here, which form different germ layers. Now we know from different lectures we have attended that different germ layers will form different kinds of organs. And when that also happens, the stemness of these germ layers is steadily decreasing. Now, we all can imagine when it comes to stem cell giving rise to a differentiated progenitors, there are lots of biochemical changes. And one of the biggest biochemical change we can imagine is the exchange of these molecules known as transcription factors. Now, when it comes to early stages of development, these transcription factors guided by morphogens forms different kinds of organs. And that's what gives rise to identity to different layers. But as the cell differentiates into further down, as shown here for intestinal stem cell, blood stem cells, or skin fibroblasts, you can imagine the kind of transcription factors giving identity to these terminal differentiated cells do not allow these cells to go back and really be stem cells. And this is where a lot of research has been focused to how to get some of these cells back to the stemness. Now, once they are formed, many organs such as bone marrow, gut, germlines, and hair follicles and muscles, how they are actually maintained, how during adult life the function is carried out, and at the same time they are repaired, is something which is very crucial, and it's something which is under active investigation. Now, what scientists have found that every of these each organs, as I mentioned, have what they call as the residual adult stem cells. And these are the cells which are specifically designed for that one particular organ. So for example, bone marrow has hematopoietic stem cells, gut has intestinal stem cells, germline has germline stem cells, muscles have, as Nag said, muscle stem cells. And each of these stem cells, which are deep seated in these tissues, can, based on the requirement, give rise to the cells required as the organ ages or there is an injury. Now, having said that, are the stem cells working by themselves? Is there any support structure which is helping them to really do their function and giving them nutrition? And here is where we introduce the concept of stem cell niche. Now, as shown here, there are two systems when it comes to development of an organ. There are the stem cells shown here on the left, which are marked in red, called as the hematopoietic stem cells. In case of hair follicle stem cells, they form bulk stem cells. And you can see these stem cells are not in isolation. They are surrounded by all these different kinds of cells, which forms niche cells. And both of these systems shows us that there are lots of different signaling molecules such as Wnt, Notch, Hippo, and many others such as BMP, which are actively playing role when it comes to stemness and maintenance of stem cell niche. And a lot of times during the aging process, there are mutations which happens, and that leads to failure of stem cell maintenance. And a lot of times that is the result of the aging process. Now, this is the question which has actually intrigued lots of scientists and forms the core of most of the industrial research and research at medical school. What exactly happens when a young person ages with time? What exactly happens at the organ level? What exactly happens at the stem cell level? What exactly happens at the nucleus level when it comes to regulation of stem cells and their gene expression? And to understand this question, a colleague of mine at Stem Cell Institute, her name is Yache, designed a model, mouse model, where you can simulate aging. Now, as you can see in this picture, there are mouse which are absolutely black in color. And then there are mouse which are white when it comes to their coat color. And this really simulates the process of aging where you begin as an absolutely brand new organism. You have black hair, but as you age, this hair kind of starts graying. And people have tried understanding how actually this process happens. In some cases, they also seen premature graying of hair. Now, what Yachi found out in her lab that there are lots of different kinds of stress which are involved in this process of aging. The more the stress, the more the graying of the hair. And as can be shown here in the schematic, what they use as a paradigm to induce this unusual amount of stress in the mouse, they use different kinds of processes. One of the easiest way to induce stress in the mouse, they have seen, if you just confine a mouse to a very tiny space, that sort of induces high levels of stress in the mouse. But to consistently get this graying of hair within the timeline by comparing age match control, they use the chemical method. And in that case, they use this very specific compound known as resinoferatoxin, which comes from this one cactus called as a Euphoria resinifera. And this is the chemical compound which is known to be 1,000 times spicier than the capsaicin which comes from chilies. Now, this compound, what Yache's lab found out, as shown here, whenever you introduce this compound under the skin of the mouse, as shown here in the blue are the hair follicle stem cells. So you are looking at the base of the hair, and the reds are actually the stem cells known as melanocyte stem cells. And the job of the stem cells is actually to form melanin required for the shaft to get its black color. Now, whenever you introduce this resin ferrotoxin, it leads to depletion of these red cells. And as you can see on the right side, whenever you do the treatment, the red label cells have disappeared. Now, this was a very interesting finding that a stress, even locally, can lead to stem cell loss. Now, they hypothesize that maybe there is something which is happening at a very specifically at a follicle level. And what they proposed was maybe there are some neurons which are actively involved in this process. And as shown here in the middle panel, there are two panels which are showing two hair follicles. There is a green thread, which is a sympathetic nervous system, which is kind of innervating the stem cell and stem cell niche marked here by TH. And what they did, they took this one particular sympathetic nerve, and what they did using this CNO, which is artificially inducing, artificially inducing excitation of these particular neurons, they show that by using this stimulus, by this fantastic system known as DREAD, which allows you to put this very small specific molecules known as clozapine N oxide. You can stimulate this one particular nerve at very one particular hair follicle. And what they found whenever they did, very similar to the toxin they were using, they could drive the stem cells out of the stem cell niche, and then it caused a depletion of the stem cells, as shown here in the plot. Now, what does this tell us? This tells us that when it comes to stem cell function, the stem cell has to undergo proliferation, but at the same time, the stem cell has to restore its self-renewal capacity. A lot of times, when it comes to stress like that, actually the stem cells do not retain the ability to self-renew. And in this case, melanocyte stem cells are actually driven out of their self-renewal status, and that's why there's a loss of stemness. Now, they wanted to understand what exactly happens when you take a stem cell niche like this and you give a continuous stimulation. What they found out, whenever you have a stress, whenever there is a sympathetic nervous system stimulation, there is a tremendous amount of noradrenaline, which is secreted as shown here in the schematic. And this noradrenaline kind of stimulates the stem cells to form more melanocytes. Now, you can imagine when it comes to a normal mouse, this has to happen with the bouts of stem cell going back to its niche, and that's why you continue to have black hair. But if you continue to give stress one after another and noradrenaline stimulation, it leads to more stem cells activated and thus leading to loss of stem cells. Now, when they did that experiment using the same toxin and looked for all the genes expressed by these activated stem cells, what they found, as shown here in the B, that most of the stem cells showed activation of cell adhesion genes, which are cadherins and other genes. Then there were a lot of genes which are involved in cell proliferation. There were lots of also genes which are involved in migration of the stem cells. there was cyclic AMP, and then obviously there were processes involved in melanin biosynthesis. Now you can see this, all the genes, these are all the genes expressed by these red stem cells as shown here. And what they found was there were no genes involved in self-renewal which were activated when you trigger stem cell activation again and again. Now the question was, if one has to rescue this phenomenon and find a drug, what is the way? So they ran a tiny chemical screen, and as shown here in the E, what they did, along with the toxin from this cactus, they also introduced some other molecules which are known to inhibit cell cycle. So one of the compound was flavoparidol. The another molecule which is involved in cell cycle inhibition was P27. And whenever they had these other molecules in the presence of constant stimulus, they saw that the stem cells were activated at the same time some of the stem cells were retained, which meant that the organism will continue to have black hair. Now, in summary, as shown here in the schematic, that when it comes to a normal mouse, it actually goes through a process of a hair regeneration, which is actually divided into three different parts: anagen, catagen, and telogen. And ideally, a stem cell has to go through all these processes. But when there is a constant stimulus, the cell, rather than going from anagen, catagen, telogen, it actually leads to continuous activation in the catagen process, and that's where the stem cells are lost, and that is what causes the graying of the hair as shown here. Okay, but if something like this happens, is there a way by which we can take a stem cell required for a specific organ and put it back and really restore the color of the hair or the organ function, and really have this organ going continuously and live really beyond 100 years? Now that kind of demands that you need to be able to take one very specific cell from an organism. It can be from the skin, it can be from your buccal cavity, and really ensure, as shown in the first video, to take it back to the stemness and really use that stemness by walking that cell back in terms of development to make whatever you need. For example, as shown here, you can make a neuron out of it, or a muscles out of it, or a liver out of it, a blood stem cell, or a bone out of it. And we all know when it comes to aging, lots of these processes are hindered. And this is something which forms a big question when it comes to stem cell biology. Now, I will stop here, and if there are any doubts, I'll be happy to answer them. Manisha, you have a doubt? Please shoot.
Yes.
Yes.
Could you just explain the last slide, the
Yes.
last part where you add some substance along with the toxin? That
Yes.
part.
Yeah. So here in E, there are 5 panels. And the first panel, which says vehicle, is essentially a mock treatment, which is inducing a syringe to introduce a saline solution. And that really says that, oh, we are doing everything when it comes to this experimental strategy. We are inducing physical injury, but there is no chemical injury, which is the actual assay, which is exactly the next panel, vehicle plus inferotoxin. Okay, now you can see between the vehicle, which is saline treatment, and the toxin treatment, the differences is the red cells have disappeared. So red cells are our stem cells, melanocyte stem cells. They have disappeared, and this is what they have been showing. Now this is where they ask, can we actually have all the stress going normally? Because you cannot avoid stress in life, you cannot avoid the exposure to the chemicals, aging process, which can cause this parasympathetic nervous system activation. Now they're saying, if that is causing graying of the hair, is there an inhibitor which can be brought in to really allow stem cells to do their job, but at the same time ensure that they are not lost completely? Now, there are many ways to do it. But how do you go about a chemical screen? Because there are thousands of chemicals when it comes to chemical screen. For that, the knowledge came from this graph, which is B. Now, this B graph tells them that whenever you give this toxin and you compare those cells with the control cells, the processes which have been upregulated are all the cell proliferation, cell division, cell migration processes. So what they did, they took these compounds 80, 7519, flavoparidol, and P27, which is the gene. Now, all these compounds and the gene P27 forms this cohort of molecules which are involved in regulation of cell proliferation. So this is what is happening. There is a signal coming from the toxin activating the neuron to tell the cells, stem cells, actively divide and proliferate. Now, at the same time, you're saying, no, we want to introduce this drug, which is flavoparidol or 80 or P27, which is inhibiting the cell proliferation. So you're ensuring there is some proliferation and then some inhibition. And this is done at the same time. It's like exposing yourself to sunlight, but at the same time having a sunscreen, which allows protection in some sense. And whenever they do that, whenever they inhibit the proliferation, they saw that the hair stays gray, hair stays black, and the stem cell continues to make this melanin producing cells. Yeah. Okay, Iqbal.
Yeah, hi Rajesh.
Okay. You say.
Manisha, sorry, you have a follow-up.
Yes, I do.
Yeah, go ahead, shoot. Sorry, Iqbal.
Yeah, that's no problem.
Yes, I was just seeing that the red cells, where the red cells were at the starting when there was just the saline thing,
Yes.
the redness has changed, like it's just shifted to some places. How is that? Like,
Yes. So, as I said, based on the stage of the hair follicle regeneration, which is anagen, catagen, and telogen, the stem cells will be in the different stages. So, anagen is when the hair follicle is actively being formed, and there are lots of stem cells which are actively forming these melanocyte-producing cells and other stem cells and other cells required for the hair follicle. And that's why the processes demand that the stem cells will be in a very specific niche, and whenever there is a requirement, oh, we need a new cell, the cell will divide and migrate to that place. So when it comes to melanin production, the stem cells divide and they go to a place where the color pigmenting cells are actually made, and that's why these cells are in the process of migrating the places where it's required. Yeah, good question.
thank
Good
you so
observation.
much. Thank
Yeah.
you so much. Yes.
Iqbal.
Can you elaborate the stresses which are affected in stem cell production?
So in this particular case, Yache, a study like this, Iqbal, takes around more than 5 years, and lots of different scientists. And they were trying to find out what exactly can be a good stress, and there were lots of different things. In case of humans, they saw that repeated, shaving can cause a lot of stress in your sclera, and that causes the stress. The other thing they were also finding out that a particular change in the pH and can cause the graying of the hair. They also found out that if you just take an animal, and this is something they were also doing on the campus, if you take an animal, it's slightly a cool thing to do. They take the mouse and put it in a bag and seal the bag. Now you can imagine the mouse is really kept in this particular space. And ideally, mouse never does that. It goes around, it explores, it really likes the stimulus. And that is known to increase the stress levels. The another thing which was slightly funny they were trying to do was, and this is very intuitive, was they were trying to take cat urine. So I don't know whether you guys know about this or not, but the cat urine smell is considered to be very stressful when it comes to mouse. And that was something they were trying to do. So there were a lot of different things. And you essentially begin with one strategy. you get some results, but to get a consistent result, you come up with a stable technology, and which is what this DREAD, which is a designer receptor system, exclusively activated by this designer drug. So you can see what happens in this is there is this neuron shown here on top on the left side, and it has this receptor which activates G alpha Q, which is cyclic AMP, if I'm not wrong. And based on this clozapine N oxide, it activates
Wow, that's interesting. Looks like he's kicked out of the Zoom. I'm pretty sure he will join in a second. Yeah.
That happened to Rajan also. He was also getting kicked out of the meeting.
Wow. I guess there is something that Zoom do not like about people in Boston. It's possible. I'm not in Boston, I'm in Cambridge, so I stayed. Yeah.
I hope the recording goes well this time.
It's still recording. I think, yeah.
Did
yes,
you
I'm-
start the recording? If you did, then that means it will still record. But if he has started, then it won't be recording still.
It says it's recording. So when I started, it was recording too. I think it was Svikruti who started the recording.
Yeah, he is back.
Yeah.
sorry, did I lose anybody? Everybody's still there?
Yeah, we
Yeah,
are here.
people were wondering if you are not going to come back.
Sorry, my laptop wasn't charging. My bad. Nagu, can you allow me to share the screen again?
Oh, yeah, absolutely. Yeah. Where is
Thank
it?
you.
Security, share screen. Yep.
Yeah. Awesome. Okay. Yeah. Okay.
There you go.
Okay. Yeah. Yes. Can you see my slides? Yeah.
Yes, we can
Yes.
see your slides.
Yeah.
Yeah.
Awesome.
Yes.
So, and so, you know,
Are you able to turn your video on, or is it not?
yes, I can turn my video. Yeah.
Ah,
Sorry.
okay. I
Forgot
mean,
about it. Okay.
no
Awesome.
stress,
Yeah. It's good to see. I have, you know, this all get up for you guys. And this sort of calcium signaling causes a trigger which leads to release of this noradrenaline as shown here. And this noradrenaline is then activating the stem cells which have receptor. Now you can understand this scientist Yache and group have found out there is a parasympathetic nervous system which innervates this bulge stem cells, and then these stem cells have receptor for noradrenaline. All of this forms one paper. So all of this work was done in one paper, and it's a remarkable feat of achievement. If you see, it made lots of news. It was covered by New York Times, and we are really proud of all of this work. It usually, whenever it comes to something like this, designing fantastic assays, you go around, give talk, and people tell there is a better way of designing the things. Yeah. Okay. So who's next? Iqbal, did I answer your question?
I have no question at all.
Okay. Who's next?
Me.
Yeah, go ahead. Sure.
Yeah. Hi, Rajesh. So my question was that I have two questions. First one was that you talked about there being over proliferation of stem cells, which loses, which then makes it like which causes the loss of stem cells. So is
Yes.
it because over proliferation causes loss in regeneration capacity of stem cells, or what is the reason?
So that's a very good question. So ideally, what happens in terms of stem cells, as Nag was saying in the beginning, that when a stem cell divides, it has the reason why the cell is called stem cell is there are two major properties. First is ability to self-renew, which means when a cell divides, it retains its stemness. So there will be two cells, and one of the cell has to be a stem cell. Okay. Now the other cell can differentiate and form the cell which we need for organ maintenance and all. Now you can imagine the kind of signal you need to do this, to go self-renew and then form another cell which can form cell we need for organ function, has to be done very carefully. Okay. Now, what is that signal? A lot of time the signal is essentially a notch signaling, a BMP signaling, or a neurotransmitter like that. Now, in that process, what happens is the quantum of the signaling decides what exactly happens to the stem cells. And in that process, a stem cell divides in a particular orientation, and the orientation is such that when a cell divides, one of the cell is pushed away from the niche, and that is what is called as the asymmetric division. Okay. Now there is another process known as symmetric division, which means when a cell divides, a mother cell forms two identical cells, and this is the process a lot of times is also involved in stem cell population right in the beginning, but at the same time can lead to differentiation of both the progenies. Now, it did give rise to more than required number of cells for making, in this case, it will be two melanocytes rather than one melanocyte stem cell and one melanocyte. In this case, continuous activation can lead to two melanocytes. In that case, that hair might be more black, but the next time when the hair comes, there will not be any melanocyte stem cell. And a lot of times, to take you out of this particular context and another context, let's say when it comes to aging, what happens is some of this stem cell signaling from the niche itself is compromised. So stem cell is still there, but the niche sort of fails to activate the signaling. And we know from muscle stem cell, it's actually delta notch signaling. The stem cells are there, but the delta which is provided by the muscle fiber is something which is not at high level, and that's why the stem cell is not activated and leads to muscle injury. So yeah.
Okay, thank you. And my other question was that this dread receptors, like,
Yes.
could you reproduce this experiment in hair follicles of other organisms, or is it just
The mouse.
exclusive to mouse?
So lots of these systems can be taken out of one organism and put into another organism. Um, and people, that's how actually most of the research is done. Once a technology is designed in one particular organism, it can be taken into another organism. Now, what particularly they have done to get this dread system in the parasympathetic nervous system specifically is they use a transcription factor driver which is specifically expressed in the parasympathetic nervous system. And right next to that, they have this gene coding for this particular dread GQ channel. And now you can understand it has only two components. It has a promoter and it has a gene which is driving it. Now you can take that promoter, let's say you want to do this in hamster. So you can take hamster specific gene, promoter, in the whichever cells you need. It can be muscle stem cells, it can be neurons, it can be epithelial cells, and then right next to it you can put this gene, and that will lead to activation of this gene in hamster skin or hamster muscles, and it should be pretty easy to do. There are a few things which you change here and there, which is a common process when it comes to standardization, but it should be doable. Yeah.
Right. And I'm sorry, I just have a follow-up.
No,
So
no, go ahead. Yeah.
So what about stress causes stem cells to render inactive? Because the thing is that if you take an organism like a mouse, which is very locomotive and it moves around everywhere, and if you lock it in a place, it becomes stressful for that
Yeah,
a particular organism. But if
yeah,
you take a particular organism like a slug or like some
yeah.
organism which does not move a lot, then even if you put it in a particular place, like lock it somewhere, it's
It
not a
wouldn't cause
stressful
any stress.
environment for that organism. So
Yeah,
that organism may not, like, I'm not saying that that organism has hair or something like that, just the stem
yeah,
cell part of it. I'm just like giving an example
yeah,
hypothetically that that if that organism has hair, then it wouldn't be gray because that's not a stressful environment for that organism because it does not
yeah,
move around a lot. So
yeah.
what about stress causes the, like, what is that trigger point?
Yeah.
Like some must be
What
certain threshold.
qualifies as a stress for different organism is very different. Like for example, when you are young, going on playground is not a stress. But if you ask Nag at this point to run few miles, it might be stressful to him. So but Nag is really good at running. So it based on
I'm very much
life.
dying. I'm not doing that at all.
So based on your situation, the definition of the stress is very different, and people are trying to find out what exactly constitutes stress, and including drinking soda water can be stressful to your intestinal stem cells, and that has underpinnings when it comes to change in the pH levels. As simple as that. So there are lots of different kind of stress. Uh, and almost everything repetitively can constitute stress, and there are many examples of that. For example, when it comes to muscles, what they are seeing, continuous activation is really good for muscle maintenance and bone density and all, but if you overdo it, it kind of drives that muscle or bone to another level where it fails to maintain. So there are many things which are considered in certain quanta good, certain quanta bad, but it's under huge investigation. Like even when it comes to cells in the culture dish, what we have seen, changing the media from one medium to another medium, which is still providing nutrition, can constitute a metabolic stress to the cells. So as simple as that, yeah.
Right, because like for humans also, like we are just so individually different, right? So like something that must
Yes,
stress you out, that might cause you to get gray hair. I might be in the same like a parameter of situation, stressful situation, but it might not cause my stem cells to render inactive. Like, you
yes,
know, so even if so you can't really, for mice maybe they are not that complex that they might have individual
yeah,
responses, but when you try to translate these studies to humans, then you can't like publish a paper saying that this is the threshold for every human on the planet, that this is the
yeah.
stress level of that causes stem cells to be inactive.
That's a very interesting point. So what has happened, what they have seen when it comes to which we will be talking about when it comes to clinical trials, uh, you bring in a drug from a research done on mouse models, and you say here is a flurperidol, and that can restore muscle stem, these melanocyte stem cells, and they're seen in the population it doesn't work for 100% of the people. It works only for maybe 10% and 12%, and people are trying to find out there is a lot of huge biological variation. Even when you say skin stem cell from one person to another person is very different, so there is a lot of biological variation. This transcription factor network is something reacts to these inputs very differently from cell to cell and organism to organism. So you have make a very good point. Yeah, thank you.
Thank you, Rajesh. Thank you.
Who's next? Yashini?
I have a
Yes,
question.
Rajesh.
You
Okay,
you say no, no. You say Yashini, you say no problem. You say.
okay. Hello, Rajesh. Actually,
Hi Yashini.
I have a couple of questions. Like,
Yeah, please.
you told about residual amount of stem cells. Like, so is the stem cell amount decided during the embryo formation, and if it is like one part of the cell will self renew and the other part will differentiate with all, like that is the only way the amount of stem cells can be kept constant. And if we assume that somehow we deplete some of these stem cells, so we cannot retrieve to the original state how it was, like the amount of stem cells that was present in the beginning. If we try to deplete it somehow,
Yeah.
the new number of stem cells and the original amount will not be equal. Is it that way? Did I get what you've told right
Yes,
about
so
that?
that's a very good question. So, if I have to summarize your questions, correct me if I'm wrong. There are two questions. First question is how the number of stem cells when it comes to each of these hair follicle or any system are decided. How do you decide it needs 2 stem cells or 5 stem cells or 10 stem cells? That's first question.
Like which
And
stage it is being formed? Like once it is formed in the embryo stage, it doesn't
yeah,
again form, like only self renuation is there, and no new
yeah,
stem cells are being formed.
yes,
That was my first part of the question. And like in some way, if we deplete the number of cells, like you told stresses,
yeah.
so in case we deplete it, the number of cells, stem cells cannot go back to its original number. Is it that way?
Yes. Okay. So now I got it. So during embryonic development, you are correct. The each of the organs get some specific number of stem cells, and that is something which forms the way the organ is designed. So every organ gets some stem cells. Now, what happens as we grow? Let's say you are an athlete, you're running around, you're going to enjoy your muscles more than other people who are actively not playing role in playing games. Now, in that case, your stem cell will be activated, they will repair, and some of the stem cells will go back to their quiescence. Now, yes, we do lose stem cells in that process. What people have seen from the research is as the organism ages, this the number of stem cells continue to decline. Now, what does that mean? That literally means if you began with 100 number of stem cells when you are 10-year-old, by the time you are 30-year-old, your number of stem cells will be used. And lots of times when the stem cell starts in the process of repair, it fails to go back to its stemness, and lots of times it just differentiates into uh, terminal differentiated cell, and they are lost. Now that is something which forms a very core interest when it comes to aging research. That why do something of this sort should happen? What they have also seen is if you just do not activate your stem cells by exercising or running around or by just using your organ regularly or abusing it other way around, then also it causes loss of stem cells. So the reason why somebody loses stem cells, there are many reasons for it, and people are still discovering what all can be the reasons. Yeah.
Okay, so like at the point when we lose all the stem cells, there is no some way to renew the
Yes.
like the injured cells, and there comes cell death, things like that.
Yes. So there is a time when during the normal process of aging, what happens is you your all the stem cells are very old. They are there are no stem cells, and that's when your organ sort of starts losing its function. And I'll come to that in the next section. Yeah.
Yeah, I have one more question. Like
Yeah.
in
Please.
in plants, there is a process of like after differentiation, there is a process
Yeah.
of de-differentiation and again
Yeah.
redifferentiation. Like I
Yes.
don't remember very good, but I think in the formation of cambium or something.
Yeah.
So is there a process like that in animals where the differentiated cells will again, like when you talked about reversible process in the
Yeah.
beginning, like is there any process that is happening in humans or animals?
Yeah.
Like
So
that?
they did see that. That's a very good question. Very exciting question. It's a field where there are lots of people with lots of different opinions. I'll be brief about it. We'll come to this again. In case of intestinal stem cell, they saw that some of these stem cells, when they form these intermediate precursors, which are not stem cell per se, can really, in the absence of stem cells, can form stem cells. So these were the cells which were forming, other intestinal cells. But if you deplete all the stem cells, these cells are dramatically capable of redifferentiating into stem cells. Okay. I'm gonna go to the next section. Everybody who has raised their hands, I want you to write your questions and ask me at the end of lecture. Okay? How does that sound? Yeah? Okay. Sorry. So here's the question then. If you can take any adult cell and really differentiate into the organ you need, can you really put this to use when it comes to organ damage and repair? And literally what I just told was done by these two scientists as shown here, Shinya Yamanaka and John Gurdon. And what they did by two different approaches as shown here in the schematic. In the first approach, John Gurdon, who's on the right side, took a fully differentiated intestinal cells, not a stem cell, cells nucleus, and put that into oocyte. And that sort of reprogrammed this nucleus, which was fully differentiated, into a more naive state. And that formed embryonic stem cells, and that could give rise to the whole organism. In case of Shinya Yamanaka, what they found out, they took these four different transcription factors, OCT4, SOX2, KLF4, and c-MYC, and they used this cocktail of transcription factors into a fully differentiated cell. And when they did that, that also changed this fully differentiated cell into more like embryonic stem cells. And that he called as induced pluripotency. Now you can imagine these were the nuclei of the fully differentiated cells, and they were pushed back to embryonic stem cell state. And from this, you can literally from every organ you can imagine, and that fetched them this Nobel Prize in 2012. Now, to tell you some details about this experiment, John Gurdon, when he was a young grad student around 1960s at Oxford, he took intestinal cells from this Xenopus frog, and when he was doing that experiment, he took cells from an albino frog. So this frog in the corner, a tiny frog, is actually albino, which does not have any skin pigmentation. He used those cells, took the nucleus out of one single intestinal cell, and took oocytes from a green-looking frog, and then removed the nucleus of that green frog oocyte and replaced that with intestinal cell nucleus, which was taken from the albino frog. And in this process, he literally got frogs which were looking albino, very similar to the first frog he used to take out the nucleus. And this experiment in a nutshell showed that what people believed till then was that if a cell takes a progressively defined state, then it cannot form other kinds of cells. What he did, it took a fully differentiated cell's nucleus and reprogrammed by doing a nuclear transfer. Now, there are lots of different animals which have been cloned till today, including sheep, mouse, cows, and pigs. And here is the result of his experiment. The center, the big frog, is actually the mother frog, and the oocytes were taken from that frog. And the rest of the frogs here are all the male frogs which were made by using the intestinal cell nucleus. And here is John Gurdon when he was young. And you can see he has a tray which has lots of frogs in it. So the interesting thing about John Gurdon is till today, although he is more than 60, 70 years old, till today he goes to lab on daily basis and does his experiments besides his, really next to his grad students and postdoc. And that is something which is remarkable when it comes to John Gurdon. There is another interesting fact about John Gurdon was when he was a young student, his teacher, biology teacher, thought that he was not capable of doing any biology, and he was literally written off that this guy will not be able to do anything, so there is no point in enrolling him. Find out that later and read what that teacher had said. And definitely at this point, John Gurdon has a Nobel Prize. So it says a lot about what you can do by just sheer determination and grit. Now, coming to Yamanaka's factor, what Yamanaka did with Kazutoshi Takahashi, who was one of the senior scientists in Shinya Yamanaka's lab in Kyoto University, They took these 24 different transition factors. Now we all know that transition factors are the molecules which bind onto genome at different locations and start lots of transcription. And lots of these transition factors have thousands of binding sites. What Takahashi did, he took 24 factors and said that maybe some of these factors will turn this fully differentiated cell back to the stem cells. So you can imagine how many number of experiments he had done. And what he found out by using combination of these different transcription factors and using mouse fibroblast, which is skin cell, he could come out with these 4 factors, which I mentioned earlier. And they could actually drive these fully differentiated cells, shown here on Waddington paradigm, back to the stem cell state. And this is something which was really remarkable when it came to the stem cell cells. Now, if you guys get time, I would highly recommend to read these 2 papers. And if you need those papers, let me know. I'll send you a copy of it. And this is something which summarizes how you can take, as Yashini was saying, a fully differentiated cell and really produce the kind of stem cells you need based on the individual requirement. As also Sharoni was mentioning that maybe based on the stress, I might be having a problem in a different organ. My hair may not be growing gray, but maybe my muscles are growing weak with my, the stress. And I really don't need mesenchymal stem cells or melanocyte stem cells. I need is what muscle stem cells. And using this technology, you can literally make those muscle stem cells. Now, having learned about human cells and what all it takes, so much effort to convert a small cell into a stem cells, there are a lot of different organisms, such as this flatworm known as planarian, which are able to regenerate whole body within less than a couple of weeks. So here is shown as a flatworm, and you can literally cut this into as many pieces as you want, and each of these small pieces can really regenerate the whole organism. Now I'm going to play a video. The most of the work we are looking at was done by Phil Newmark, Alejandro Sanchez Alvarado, and Peter Reddin, who is here at MIT. So if you get time, do visit those labs and see what they have done. And here is the video. And in the video, we'll be seeing how this one tiny flatworm, planarian, cut into 2 pieces, the tail and head piece, can regenerate full organism in less than 7 days. So, so what they found out actually during this process, as you can see on the screen, there is one tiny, very tiny white looking tissue. And what they found out from the work of Alejandro and Peter Reddin and Phil Newmark, all these scientists found out there are these very, very tiny stem cells in the planaria known as neoblast. And these are the cells which are, by definition, what one can call it as a pluripotent stem cells, can give rise to the every possible tissue when it comes to planaria. And the kind of signaling it receives from the whole body is something which is under investigation. Now, we can learn a great deal of biology and regeneration from an organism like this. But as Sarwar mentioned, that lots of the understanding which will develop from an organism like this may or may not be translatable to the whole organism, such I mean, slightly higher organisms such as mouse or even humans. But a lot of times you understand fundamentals of stem cells by studying on the simpler organism. And this is where it all begins. A lot of different times a chemical screen is done on these molecules. A lot of different times a genetic screen is done on these molecules, which allows you to do everything within 7 days. And you can imagine doing the same experiment on the mouse, which will take years. So you can literally finish the whole screen. This organism is literally something which can be kept in a very, very tiny dishes. And you can run the whole chemical screen in less than a few months and really have a good target. And a lot of times that's how the model organisms are chosen. So with that, I would like to thank Nath and Vinay for giving me this opportunity, and the Lakshmi Mittal Family Foundation for supporting their adventure, and all of you for being such a great student and asking fantastic questions. And with that, I will stop, and I would like to go back to the questions if you guys have any. Thank you. So, Shirvari, I will come back to you later. I already took some of your questions. Akhila, why don't you shoot? Akhila, Shreya, Shumoi, Chamundeshwari, anybody?
Hi, Rajesh.
Yes, hello.
As we know that stem cells are generally stored under low temperature and they retain their capacity, right?
Yeah.
So how
Clarification.
do they
Yes.
store at low temperature? Does low temperature or high temperature does not affect the stem cells?
So what they have seen is in case of metabolism. So I guess you're referring to cryopreservation process. Am I correct? Yes. So when I do my experiments on hematopoietic stem cells, these cells actually come as a very tiny vial, and the temperatures are below, minus 100 or so. Now, what they called as that state as a cryosleep. And what cryosleep does is it actually sort of slows down every process of metabolism, which is production of ATP and then making proteins, making RNA. And there is a specific way this is done. So the way it is done is you take a stem cell and it is usually surrounded by lots of this rich nutrient medium. You can think of it as a very nice soup, and that is what keeps them alive. But when you want to put them to sleep, what you do is you replace that nice soup with another compound known as DMSO, dimethyl sulfoxide, and which is something allows these cells to go into cryosleep without rupturing because of the freezing of ice molecules. So what you do is you put a lot of DMSO and then some of these rich nutrient soup, and then take these cells, spin them down, and keep them in the -80 in a very special container. What this container does is decreases temperature 1 degree Celsius at a time at a very slow rate. So it's literally like putting them to sleep by singing a lullaby, and then you stick them into the liquid nitrogen. Now, all this process literally slows down the jiggling of biomolecules, and also active translation and active, all of those processes which are resultant because of the Brownian motion. And that's how they are stored. Now, to take that the same analogy in terms of temperature change to model organism, lots of times stem cells in the body are also kept in those specific niches I mentioned, where there is a lot of differences when it comes to the oxygen levels, the kind of molecules comes in, the kind of CO2 in it, the pH of the particular area, and lots of those things. So there are lots of very specific things which are different when it comes to stem cells in comparison to differentiated cells. Now, if you want to take those cells back from the cryopreservation into normal growth media, let's say you want to put them into the patient, then you do exactly other way around. Then you thaw the cells, you remove the DMSO, you spin them down, and you give them new media. And that also takes around 24 hours for them to come out. Good question. Who wants to
One
go
more
next?
question is
Yes.
that if people live in two different environment, like one is in India, other was in US, then
Yeah.
what about the stem cell count in them? Does any variation?
So what people have seen, actually based on where you live and your nutrition and in general your lifestyle, has a great impact on your longevity. Now, if I have to take the same thing, the longevity, and draw a correlation with the organ maintenance and tissue repair, then you can see there are a lot of times when you live in a country where the standard of living is better, the pollution is low, or the temperatures are low, or the food habits are different. There are some differences in terms of stem cells. What they have seen in terms of muscle mass, what they've seen in terms of, in general, the habit of exercise and all, those are the things which really impact how actually being in different country affects you. So that necessarily doesn't mean that if you're in different continent, you will be different. What happens is when you go to a different place, everything about you changes. The temperature changes, your food habit change, your work style changes, your lifestyle changes, there is less pollution, the sunlight changes. So there are a lot of complex factors which come in, and they do impact, the kind of stem cells you have. Like for example, when it comes to your skin color and the propensity of anybody getting skin cancer, it dramatically varies if you have a lighter skin color. And if you're lighter skin color, you're gonna get more skin cancer. As simple as that. So there is some relationship. They are also seen in case of prostate cancer. In case of males, there is a high propensity of males in the United States to get prostate cancer, and that has a relationship with the processed food. So if you eat a lot of processed food, it has compounds which causes some of these cancers, which is also probably because of some stem cells going wrong. Yeah, good
Rajesh,
question.
one more question.
Yeah, sure, go ahead.
You said that there are certain chemical inhibitors like flavoperidol and P27 that
Yes.
they inhibit the cell cycle in stem cells, right?
Yep.
So at what stage of the cell cycle the stem cells are arrested? I mean, what stage of cell cycle is arrested in stem cells that so
Got
they
it.
can retain the capacity to
Stem cells.
regenerate
Yeah.
at totopotency?
Yeah, that's a very good question. That's a very, very, very good question. Just to divert you a little bit from the question, this same question was asked by the lab of Thomas Orlando, who's at Stanford, and he works on muscle stem cells. One of my heroes when it comes to muscle stem cells, and his lab produced this fantastic result. They found out that if you injure muscles on the left side of the body, those stem cells are activated when it comes to muscles. But they also found out the stem cells on the right side of the body, or vice versa, were also activated. And they found out that lots of times the stem cells, when they are quiescent, to answer your question, Akila, they are in G0 state. Okay, and that's the stage where they are actually sitting quiet and waiting for the signal. What they found out, when you injure cells on the left side, those cells rapidly get into S phase and G1S transition, and whole stem cell get activated and gets into cell cycle, actively proliferating and making new proteins and all of it. But that injury also kind of activates stem cells on the right side, and they are not necessarily in G0 stage. They get into this middle stage, which is known as G alert. And that's a very fantastic finding. You might want to dig up more on it. And they found out that when the cell is in the G alert stage, if you give little nudge on the right side, the time it takes to repair muscle is less. So you can always think of these cells are ready to go. They are more prepared than the G0 stage because they are already in G alert state. Yeah. Okay, that was a good question. Halak.
Thank you, Rajesh.
Yeah, welcome.
Yes, hello. I have a
Yes,
question. Yeah. So I have a question that you talked about the designer drugs. So how
Yeah.
are they? What
Designer
specifications
drugs?
do you need and how do they made?
How do they?
Are made?
How are they made? Okay.
What
So
perspective you need to talk? Yeah.
very good question. Very good question. So what happens is whenever you want to design a specific drug, these days the process has been slightly simplified because of the work by this guy. He's the big lab in Seattle. His name is Baker. And you might want to find out what all he does. So he works on protein design, and a lot of different times designing and understanding protein structure is a big job because of the amino acids, how they are organized. He had come up with an interesting algorithm which is based on GN Ramchandran's plot, which allows specific steric hindrance based on uh, the kind of atoms you have, the kind of amide bond, and the secondary chains. And what you do really when you are designing a drug is you actually have a protein structure in place by using simulations. And then you have a chemical library, which is also virtual. And what you do is you throw in this chemical library in the computer onto those structures. Now, in this case, it was a membrane receptor. And what you're really looking at is a compound in the computer binding to a protein in the computer with a minimum energy, which is something which happens in the natural life. Now, lots of these compounds have similarity to the natural structure it binds to. So, for example, let's say if I want to find antipsychotics, then I need to know what neurotransmitter look like, and then design a structure which is similar to that, and just very few chains, and really find out that it binds or not. So it is a long, lengthy process. It involves lots of collaboration, lots of understanding of structure and chemical nature of each molecule, and also how water molecules are sort of organized around protein molecules and other simpler compounds. Good question.
Thank you. And are there any specific softwares used in them, like Cassava or something?
Physical structures?
Online software, specialized software.
Online softwares, yeah. So there are lots of softwares. There are two which were published recently. It might be slightly hard to digest at this point, but you can go and look for AlphaFold.
Thank you.
Awesome. Who's next? Shumoi?
Yeah, so before I ask my questions, I wanted to ask that Halak asked about this, like this drug designing and all. So does that involve de novo protein designing, which you talked about? You talked about Baker, right? So does that involve de novo protein designing?
It does. So a lot of times what happens is the de novo protein design is really based on what GN Ramchandran says what is allowed when it comes to a protein structure. And it's a slightly complicated plot. Maybe some of you can take it as an exercise. It's based on, when it comes to a structure of a protein, there are only basic um, units, which is alpha fold, beta sheet, beta turn, and then unstructured regions. And a lot of these structures are possible only when you follow certain rules, which is based on G. N. Ramchandran. So if you want to make a protein which is very similar to, let's say, a serotonin receptor, then you need to have all the structures which will allow this protein to be stable in the cell membrane. And it does involve lots of knowledge from the previously published literature. So even if you're designing an altogether new protein, a lot of it is based on what's known from individual amino acid properties. Having said that, you can have artificial amino acids and make altogether different proteins, but they will still follow to certain extent what G. N. Ramchandran has said, because there is a chemical structure to it and it has to follow the laws of chemistry.
Okay. So
Yeah.
my next question goes like, when you said that the stems so do the stem cells undergo apoptosis, or they undergo apoptosis after terminal differentiation?
Stem cells, they have seen lots of times, based on what exactly happens during injury process or infection process, or in general when it comes to a sort of fault in the self renewal. The stem cells do undergo apoptosis. Um, that can be one of the reasons why some of us cannot regenerate like planaria, that the stem cells after a while they are just disappeared, and they are either fully differentiated or they undergo apoptosis. Like for example, when it comes to your hand, in the beginning your hand is a lot like a flap. The reason why you have fingers, there are lots of cells which were asked to die, and that's how you get this form, which is very useful when it comes to grabbing onto things. So it's a very regulated process, but a lot of times when there is unusual amount of stress or an injury or chemical injury, cells do undergo apoptosis, and that is something people are trying to find out, what exactly causes stem cells to undergo apoptosis, and can you avoid that. In the lab, at this point, we have a project on this ribosomal protein known as RPS19. And what they have found, people who have mutation in RPS19, which forms a very important part of eukaryotic ribosome, at least in case of higher mammals. Mutation in this gene, when you have one copy mutation in this one gene, it leads to diamond blackfan anemia. Now, what happens in diamond blackfan anemia is, as name suggests, there are less number of red cells which are formed, and that leads to your body having less oxygen. And in general, the life experience has changed. So, if you have an injury and if your blood, you lose a lot of blood, there is a good chance that you might succumb to death, and also it leads to a lot of other different defects. So, what we are trying to understand is, whenever there is a mutation in RPS19, those cells undergo apoptosis. So if you start with 100 cells, 100 stem cells, and if you get a mutation in the, let's say 50% of those cells in RPS19, which is a ribosomal protein, as I said, then lots of those cells will just die natural death because of apoptosis. And our lab found out that if you play around levels of P53, again, which is a cell cycle regulator and cell survival gene, then you can allow the cells to really live longer and restore the levels of red blood cell. Yeah?
Yeah.
And I'll be talking about it maybe in the third lecture.
And I have two more questions. Like, since I come from physiology background and we are taught sports physiology, okay, as well.
Nice.
So I was asking that muscles contain stem cells, right? So during this, like the practice and everything of an athlete, so they go through a rigorous training process. So does that training process somewhat induce like the threshold of activation as well as the deactivation of the stem cells?
So what they have found in case of athletes or in general any individual who engages in physical activities, their stem cells, there are these micro fractures which occurred in the muscles. And those micro fractures are good enough signal for muscle stem cells, also known as satellite cells when it comes to higher vertebrates. And that is good enough signal for them to be activated. Now, that what does that mean? It means that if a human being who's not physically active, and if you compare that with a person who's physically active, the kind of response you will have after the injury will be dramatically different for the person uh, who's athletically, engaging in the physical activities. What they have seen, what changes when you actually physically take part in this strenuous processes is there are a lot of neurons and the tendons and supporting cells and the basement membrane, the muscle lamina. It is in close contact with satellite cells. It literally sits on top of this basal lamina, and that is in constant flux with the different chemicals between the muscle fibers. And you can imagine a muscle which is active has a different amount of flux between these cells, and hence the activation is really different. That's what they have seen in case of people who are actively involved in athlete. The number of active stem cells based on the levels of PAX and MyoD are really different. And the process of they activating MyoD, which is the next transition factor right after PAX3, which is involved in myoblast formation and the fusion of the muscles, which I'll be talking in the lecture 2. So stay tuned for it.
Okay, and my last question is in case of choriocarcinoma, where the placenta, a part of placenta stays in the woman's body, like after giving birth, that
Yeah.
that actually is cancer. That's choriocarcinoma. So so placenta has like some part of the umbilical cord as well. Umbilical cord has the stem cells, right? So
Yeah.
does that carcinoma result from the stem cells that that is left behind inside the woman's body? It means this stem cell responsible for this kind of cancer.
Yeah. So all the processes involved in terms of birth, the umbilical cord and the placenta, what they have seen, it has tremendous potential in terms of all sorts of cell lineages. Now, all of these processes have their timing. Like for example, when it comes to umbilical cord, it has the function and it is involved in active process of repair when it comes to fetus and all. But after a time, it has to disappear, and the program for their death is actually encoded in the signaling pathways. Now, a lot of times, what people have seen, the reason why there is a cancer, it's slightly vague and general explanation. Whenever there is a process, high frequency of mutation leading to cancer, they have found that there are some of the cells which are not fully differentiated, and those cells are capable of changing into stem cell like state, and that can be the result. Those cells can be the reason behind the cancer, but a lot of times the cancer can arise because of lots of different kinds of cells taking out lots of different kinds of it. So having said that, I'm not the expert when it comes to this one particular tissue, but my understanding from different kinds of tissues is that if there is a cell which was capable of forming different kinds of cells, you can literally induce one gene like a c-MYC into that cell, and that cell will become cancerous. A c-MYC change levels can dramatically change any cell into a highly proliferating cell. And that's one of the molecules Shinya Yamanaka used to reprogram, fully differentiated cell into a stem cell. Okay. Who's next? Is there anybody who has one question? I want to take people with one question. Okay, Iqbal, I have already given you one question. Who else? Is there anybody? Okay, Iqbal, you can shoot.
I have a question.
Yes, who's this? Shreya
This is
Chamundeshwar?
Shreya Chatterjee.
Shreya, go ahead.
Okay, so today I was learning a little bit about the infestation of H. pylori in the human intestine. And as you said, for the intestinal stem cells, having a lot of soda water can also cause its
Stress levels.
yes. So how can one understand if something is stressful for them in a generalized way so that we can come up with a paper that can say what are the things that can actually decrease the effectiveness of the stem cells?
Very good question. So, what people have found when it comes to the changes in the stress levels of any cell, there are few things which change, and one of the things which changes dramatically is ROS levels, which is reactive oxygen species, which can be a resultant of the mitochondrial activity. And that's just one of the ways you can design a reporter. And if you have a reporter for ROS levels in the cell, you can take control cell which has a normal levels of ROS, and then take stem cell which where you're throwing on different kinds of chemicals to study stress levels. So all you need to see is the levels of ROS going up and down. Similar to the gene levels, these people, Yachi's lab had seen. You must remember that there was this graph where they looked at all the genes which were upregulated when the toxins were added onto these hair follicle stem cells. And people are finding out lots of different reporters, and they have made the job very easy. But the definition of stress itself is very fluid at this point. What constitutes stress? Some people say ROS is good, some people say ROS is bad, and based on cell status also, it depends what you call it as a stress. But yes, there are some consensus when it comes to the stress levels. There are lots of different things which also change. For example, if you see a nucleus and the genes on the nucleus, there are different places where on the histone, different chemical moieties are added. And those are known as methylation, acetylation, sumylation, phosphorylation. And these all changes in the what they call as epigenetic modifications are also come under definition of how to constitute a stress. And people are seeing those are the changes which matters when it comes to a stem cell stemness. So there is a lot of literature on it. Very good question, Shreya.
Thank you.
Yeah, Chamundeshwar.
Hello. Yeah. Hello, Rajesh. Can you hear me?
Yes.
Hello. I have two questions. Like my first question is, like I have heard that the nerve cells, whenever some nerve cells damage, they don't have the capacity to regenerate. Oh, are there any evidences that the stem cells regeneration is limited in certain cases?
Yes, that's your first question. What's your second question?
My second question is whenever we can see that in the during the bacillus stage, that is when the embryonic cells and stem cells can be actually seen, they would give rise to like organs and organ system they would like to develop. And we can see some people they will have like one lung, one kidney. So it is something which is dependent on the genetic cell line, or is it something that the stem cell is unable to produce that kind of organs?
Yeah. So the first question is when it comes to a neuron in an adult, and if you injure a neuron, really snap it into two, can it regenerate? Now, what they have seen in case of axolotls and other organisms, it actually can regenerate. And the way it regenerates is there is a neural stem cell or there is another stem cell like neoblast, which can form the whole neuron again. What they have seen in case of higher vertebrates is whenever there is a neuron which has some injury or damage, there are these cells known as glial cells, which are supposed to do the similar job. And they sort of forms the stem cells of the brain. With age, these glial cells lose their capacity to self-renew and repair. Now that's first reason. The second reason is whenever there is a damage, it's actually made worse by some of the immune system which is in the brain, and that immune system attacks these neurons and actually makes the matter worse. So those are the two reasons why humans cannot really regenerate, huge amount of neurons. And there's a lot of literature going on how that can be, reactivated, and can you really regenerate the neurons. Having said that, people have been using ips lines to take a skin cell from the same individual and do allogeneic transplant, which is differentiating a cell into a neuron and using the whole neuron, which will be transplanted into that one particular tissue, let's say spinal cord, if there is a spinal cord injury, and really allow it to heal. And a lot of those cues, what kind of cell it needs to be formed, are given by local signaling. And local signaling itself tells, oh, we want this unipolar or bipolar neuron in general. Your second question was, there are individuals who do not have same number of organs as a normal individual, and what causes that? Now, there is a process in development known as canalization. I don't know whether you guys have heard about it or not. Canalization is a process where there are tiny variations during development, and a stem cell will, or an organism or germ layer will continue to go through development and will form even in the odds, in the face of odds, the same tissue, and it will be fully functional. Now, why some people have one kidney, why some people have a tinier lung or a smaller pancreas? There are a lot of different things which has to go clockwork when it comes to development. There are morphogens which are guiding transcription factors, which are also getting inputs from lots of signaling pathways. And if one of the major transcription factor is not activated at the same level or at the similar level or at the same time, then defects like these are seen. There is a whole biology of teratoma, which is early developmental cancers, where you see a ball of cancer which literally has all the germ layers which can form all sorts of organs. And here we are looking at cells which are very similar to embryonic stem cells, but they do not form organs. And people are trying to study a lot of that. When Shinya Yamanaka, to take some detour from what you asked, Shinya Yamanaka took those ips lines and grafted them in the mouse skin. They found out that the same teratomas were formed, which is essentially these tumors which have all the germ layers and cannot form a normal organ. But when he took the same cells and put them in the early developmental stages in the embryo, when embryonic stem cells are sort of giving rise to organs slightly later stages, they could form a really nice organ. So you're getting it. The same cell which is forming a cancer can really form the whole organism because it has all the potential, but the signals coming from neighboring tissues are slightly compromised. So lots of niche signaling decides what happens to a stem cell. So if the niche is compromised, the stem cell function is compromised, and it is actually hand in hand. The process has to go well. That's why when it comes to pregnancy and early developmental stages, you are advised not to smoke or drink alcohol, take weird drugs, aspirin, and lots of different things, which can have minor effect on the developing embryo, developing stem cells. Yeah.
Okay, Rajesh, thank you.
Yeah, you might want to read about the cyclops sheeps, which have only one single eye because of inhibition of hedgehog pathway. They look amazing.
Oh yeah, sure, I'll go through it.
Yeah, it's amazing to study. It comes from one of the plant which these sheep or goats eat during grazing, and then they found out that just grazing onto this one weird plant leads to inhibition of hedgehog pathway, and the babies born have single eye. Yeah, right in the middle, like Shankara. Yeah. Okay, who's next? Who has one question? Iqbal?
I have one question.
No, I think I'm going to go to Iqbal and then come to you. Yeah.
Thanks, Dr. Rajesh. I have a random question at all.
Yeah.
is it possible to transformation of different kind of cells like muscle cells to nerve cells or anything else have any role of the stem cell if it is possible?
Yeah. So what they have found using IPS lines, using these 4 transcription factors, c-MYC, KLF4, and 2 others, which I just mentioned. Uh, you can literally drive any cell into embryonic stem cells. KLF4, like for example, nano was one of them, which they found out was not something really required. So these are the 4 transcription factors: OCT4, SOX2, KLF4, and c-MYC. And you can take these factors, overexpress in any kind of cell, as you mentioned, and really drive it back to the embryonic stem cells. Now, in this process, they also found out that some cells are more efficient when it comes to forming embryonic stem cells. And when they were forming, they still retain some of the original characters. So, for example, even when they were stem cells, they still had some of the genes active from their muscle, memory. They were still expressing some of the myosin for the muscles and all. And the reason why they found out that these 4 genes, what they do is they go and really change the way the genome is organized. But some part of the genome is stubborn, and it says, I'm not going to change, I'm going to continue to do what I was doing. And that is something people are now trying to work out, how to make more efficient transformation of any cell. Like for example, you really want to take cells which are easy to operate, which will not cause you trouble, like buccal cavity. You take a swab and you give that cell, and you have a new muscle or a new liver ready. But then they saw that some of the cells are not the best cells, and you can think of reasons such as telomere length. If the telomere length is not good enough, then the new organ which is given to you has maybe 6 months half-life, and then you need another organ from 6 months from now. So those are the different things which people consider when choosing the cell. Like for example, when Gordon took intestinal stem cells, the reason why he took intestinal cells, not stem cells, intestinal cells, because these cells were very large. The nucleus are very big. So if you want to take out a big nucleus using a very tiny micropipette, it's very easy to do. But if you take it from a blood cell which has a very small nucleus, then it's a difficult experiment to do. So lots of times you choose a tissue and the cell based on what you really want to do. Yeah. Okay, actually, yeah, sorry, Mittal, go ahead.
Is it possible for human cells?
Yes, so what people have done recently, so IPS lines, people have used human cells, and they have made human cells into different organs. So it is possible, the same technology IPS, and the Kyoto University is actually at the front line of making all of this. What they have also done very recently is they took a pig and they created a knockout of pig. Now you can imagine a pig which is a knockout for genes involved in kidney development. So this pig can develop fully, but it will not have kidney. And what they did in that pig was they took IPS lines generated by Shinya Yamanaka's 4-factor technique, and they put those cells, just cells, human cells, IPS lines, into early embryonic stages. And then what they found, the whole pig is pig, but the kidney comes from human cells. So now you have a kidney ready-made for you in the pig. So you can just take it out and stick it in a human being. Yeah, it's pretty cool. You might want to read about that. Yeah, it's a very cool experiment.
Yeah.
Okay, Lakshmi, your turn.
Yes, Rajesh. Like, how does the stem cell actually look like, and what does it contain? Like, it has only the nucleus, and it is arrested in the quiescent phase. And
Yeah.
when it is activated, all the other cell organelles develop as per the need of the organ. Or like for your RBCs, we don't need, like in the early stages we have the nucleus, and later it doesn't have a nucleus. Likewise, when the stem cell develops, initially it has only does it only have nucleus, and based on the need it develops its organelles, or does it already have the other organelles in it within the
So
cell?
what they have seen based on, if you see early oocyte, Then the early oocyte gets half of the chromosomes from a father, half of the chromosomes from the mother. Now, here is a question. Uh, when the oocyte was formed, did it have all the organelles? No.
Yes.
Uh, you know, it had almost every organelle, but it did not have half of the nucleus, right? Half of the nucleus, half of the chromosome came from mother. And if you say, oh, when the sperm fused, what happened to its all organelles? Now you can imagine the mitochondria only comes from oocyte. So all the mitochondria we all have comes from mother. It's that simple as that. So that's the case of the oocyte, which forms, early stem cells. But when it comes to adult stem cells, they have all the organs. Now, what is the difference between, a perfectly formed terminally differentiated cell like there's the intestinal cell and early oocyte, and that is something which is remarkable of interest to lots of scientists. What they found out that these cytoplasm, which is what in this case at least is the game changer, it entrains this nucleus which is fully differentiated from stem cell, is this cytoplasm has all the organelles and this very specific histones which are involved in stem cells. And these histones are something which go and then bind to this fully differentiated cell's nucleus and then turn that into stem cell nucleus. There are a lot of epigenetic modifiers also sitting in the cytoplasm, and that is what keeps this cell as a stem cell. That's a very good question. So it has all the organelles, but in addition to that, it has the special things which keep the cells in the stem state and keeps the whole genome sort of open for to take intestinal state, stems, muscle state, or it can be follicle state. And once that decided, the rest of the regions are sort of locked down. You getting it? It's like entering into a palace and really going for one room and then closing all other doors. It's as simple as that. And then the histones, what they do is they keep all the doors open. The moment you want to take a fate, the histones are removed from those regions and new histones are placed, which will close those doors down.
So,
Yeah.
like when Sumoy talked about apoptosis, like if a cell has to undergo programmed cell death, it should have certain kind of regulatory mechanisms that
Yeah.
happen within it, and like until it is in the quiescent phase, the stem cell will not have that function of going into the stage of apoptosis, or like when will it gain that functionality, like for that to undergo apoptosis, or
So what happens is lot of times you have to understand a cell is not designed to die. Okay, cell's whole job is to survive and divide. And that's why you see emergence of cancers once in a while. Even when the cell has gone rogue, it continues to survive and proliferate. But then cells have this inbuilt mechanism which have these MHC molecules. And whenever MHC molecule takes onto this antigen, which is it's not supposed to be, that is served as a signal for a cell to die, and the immune cells comes and induces apoptosis. What triggers that? There are many triggers, and one of the best trigger known is mutation or a DNA damage. And the DNA damage induces expression of UVA, UVB genes. And when the recombination, something of that sort fails, that leads to activation of a lot of DNAs, which are the enzymes involved in chewing of DNA. And once that starts, there are lots of these molecules which are also activating a lot of mitochondria based signaling, which you guys might have studied a little bit from different lectures. And once that starts, they sit on to the mitochondria and trigger all the complement factor, and those cause lots of nucleation of lots of factors involved in apoptosis. So there are lots of caspases and other enzymes involved in this process. There are lots of RNA degrading enzymes which are also released. And once that starts, they also chew upon everything. So there are a lot of ways to reach apoptosis. The trigger can be as simple as cell failing to differentiate properly, or failing to separate chromosomes properly, or really distributing cell organelles properly, and anything of this sort can constitute a stress leading to apoptosis.
So is it possible for all this to happen in the quiescent phase itself, or like
Yes,
it
so a
happens
lot of
in the
times,
infant
yes,
phase?
it can happen in quiescent state, that the cell is just about to come out of the quiescence and it dies out. Let's say in a quiescent state, for example, if you do not give the cell, now you can imagine quiescent state is not where a cell is not doing anything. It is
Yes,
actually still
exactly.
consuming a lot of glucose. It's like you taking a tiny nap in the afternoon. You're sleeping, you're still breathing, you're using what you ate. And if you remove the glucose supply of this cell, then the cell has nowhere to go. It has to die. So that is possible.
Thank you,
Okay,
Ramesh.
Shalvari, your turn. Sorry to make you wait so long. People have 5 questions.
It's okay. Just like I would like to mention that you've used great pictures of Nag and Vinay on the
Yeah,
presentation. My friends are not that considerate while using my pictures.
that's
So
fine. I have handsome friends. I don't know about your friends.
yeah, you're a great friend. And my question would be that you mentioned that there are the mother cell gives rise to two cells, one which regenerates into some other in need, like basically whatever you need, whichever cell it needs to be, and the other one stays as a stem cell, right? So it stays in the G alert phase, and the second one it transforms into whatever it needs to be. So
Yes.
so I wanted to ask that how is that decision made, that which one will be transformed and which one will stay as a stem cell? Is it because the one cell has like lost is less capable of staying in the G alert transient state than the other cell, or is it just totally dependent on chance or something?
Yeah. So what we found out, uh, based on lots of different types of stem cell and niche context. When it comes to Drosophila brain development, what they found, there is a stem cell known as a neuroblast, and it has a very peculiar orientation when it comes to stem cell division. And what they found out is, if a cell divides such that one of the cell maintains contact with the niche, then the progeny, which is away from the niche, um, then differentiates into a glial cell or the neuron cell. And that's one context, which is orienting, division of the axis. What they found out in case of other stem cell niche, such as muscle stem cell niche, is a lot of these decisions are what they call as random, or there is a stochasticity. And what that means is, based on the stochasticity, one of the cell will become a stem cell. What we found, the underlying differences between the stochasticity are many times levels of transcription factors. So stem cell becomes a stem cell based on the levels of notch signaling. Now, if I have a high notch in one cell, the other cell immediately becomes low notch. And the high notch cell sort of tells this another cell that, oh, you're gonna be, muscles, and I'm gonna be the stem cell. And it's an instantaneous process. And there are lots of signaling, such as notch and numb signaling, which is involved in it. And I'll be talking about it in my second lecture. So stay tuned for it.
Right. And also, so if I, for example, take there's a white haired mouse and a black haired mouse, okay, and if I
Yeah.
take stem cells from the black haired mouse hair follicles, and if I transfer them in the white haired mouse hair follicles, then would they read the surrounding environment and then produce white, the shaft as white colored, or would they still produce black colored hair?
Yeah. So the people, that's a very good question. At the Harvard Stem Cell Institute, there are lots of interesting labs which are working on this model. There's a lab of Rubin Lee. You guys might want to check it out. Uh, they do this experiment on what they call as a conjoint mouse, which is essentially two mouse where the circulatory system is sort of connected. And what they did, they looked at when they did this experiment between an old mouse and a young mouse, what they saw, the factors from the blood of young mouse can actually go into the old mouse and really do repair of the old mouse. So what does that say? It says that there are lots of different things in the old mouse which can be cured by young mouse, and the factors can be the blood, can be the stem cells, and lots of things. So yes, it is possible that the factors, stem cells, and signaling molecules from a young mouse, in this case, a mouse with black hair, can actually go and change. Now, having said that, when Yache did this experiment, the levels of noradrenaline released by the neuron was something which was causing graying of hair. So it can just so happen when you will put new stem cells. For a moment, they will be able to form black hair, but if you continue to give the stress, those cells will be also driven out of the stem cell niche, and then it will become white hair again.
I
Good
see.
question.
And you talked about the regeneration of planaria and the pluripotent cells that surround it. So,
Yeah.
so Tarun took a lecture, like I guess a day or two ago, where he also talked about planaria and also how there are pluripotent cells present on the border of that organism, like everywhere basically, which can
Yeah.
just regenerate. So,
They
and
will.
he talked about there being a BMP pathway which is very necessary for the orientation of the regeneration. If the BMP
Yes.
pathway is not present, then the organism cannot tell its left from its right,
Yes.
and regeneration is faulty. So, is the BMP pathway necessary for the stem cells to recognize like its surroundings? Basically, like if I'm at the left side, so I need to produce basically the left part of the. So, does
Yeah.
it work with the stem cells, or is there
Yeah.
some like other thing involved?
So BMP stands for bone morphogenetic protein, and there is a lot of literature on how BMP is involved in early patterning of epithelium when it comes to the wing formation of the Drosophila. And what they found out is these molecules, based on their gradient concentration levels, activate certain level of transcriptional network. So if you have 100 molecules of BMP, you will activate one transcriptional network. If you have 50, you will activate different kind of network. And those differences are actually something which gives cell a sense of where they are in the whole organism. Now, the cell does not have eyes, cell does not have ears, cell does not have hands. The only way it can really sense its surrounding is by these pathways. Now, BMP is just one of such a signaling pathway. What Peter Redin, who's at MIT here, who worked with Alejandro Sanchez Alvarado, who's at Stowers Institute, found out that when it comes to regeneration in case of planaria, it's actually muscles which really lay down the landmarks. And if you compromise muscles when the regeneration happens, as shown here in the slide, then the regeneration is kind of faulty. So BMP is one of the pathways. What exactly it does when it comes to telling neoblast is something which is actively being investigated. So there are lots of different things which are being, which people say that they are important. So when it comes to this particular organism, there are like thousands of neoblasts. The stem cells are in plenty. It seems every fourth or fifth cell in this organism is a stem cell. Now imagine that kind of stem cell population. Yeah, very good question. Okay,
Yeah, thank
I
you.
guess there will be, yeah, sure, sorry.
I have one last question.
Yeah, I'm gonna
I'm
charge you guys
sorry.
$100 for
Yeah.
every question.
I'm sorry.
Awesome, good.
Saying that, so you talk about like how are stem cells actually transported from like one place to another, because there are certain so most of cells are transported in cold storage, like for the lack of a better word.
Yes.
For example, lyophilization is a technique which is used for transportation of cells, but that technique requires like freezing and vacuum, a creation of
Yeah.
vacuum. So that must hinder the properties of regeneration in stem cells
It does.
if such hard. Yeah, so how are they are transported without in getting contaminated or something?
So what, it's something, it's a very challenging biological problem at this point. And I don't know whether you guys, any of you have heard of this banks where you can really store the umbilical cord for a meager amount of 50,000. And that is now, common practice in India. And they say that that is the best gift you can give to your kid, that you give them umbilical cord. And the reason why they say so is they say that umbilical cord has, as you all know, the stem cells which can form all sorts of, kind of cells, and they are very new, naive, and all. There is hardly any DNA damage. But when it comes to this cryopreservation, the technology involved is not fully yet there, and there is a lot of loss involved. Now, the question is why we do not know how to exactly do it is because our understanding in terms of how cell behaves, at fundamental level, at mitochondrial level, at endoplasmic reticulum level, at nuclear level, is very limited. Though there is a great deal of biology we know, and people are trying to find out how to do it. There was a very interesting study about how to preserve Drosophila larvae, and surprisingly, what they found out, you can really throw Drosophila larvae into cold storage at -20 and resurrect them after 6 months. You can just take them out of the freezer and they will be good enough to go. And what they found out, to allow you to do that, you need to feed them a specific protein-rich diet before you throw them into the cold storage. Now, you can understand probably there is a signaling pathway which is altered when you feed them high protein diet and all. But a lot of literature, there is a lot more research needs to be done. There is one specific stage in case of C. elegans known as Down stage. You guys might want to read about that as well, which is also very similar stage where it's a quiescent. So if you take away the nutrition, the C. elegans kind of goes into this cryo sleep without doing much, and it stays alive without any reason, without any support. And in that case, you don't even have to do a lot of cryopreservation. So we are still learning about it. There is very interesting scientist here, George Church. He's trying to get these mammoth cells, a nucleus, and put that into elephant. You might want to read about that too. How he's planning to resurrect woolly mammoth. It's an exciting project. Yeah, using elephant oocytes, a lot similar to what John Gurdon did.
Right. Okay. Thank you so much. Thank you.
Awesome. Okay. More questions. I would like to take more questions later on. Nag has some announcement to make. What you can do is email me those questions or have them ready for the next talk, and then we'll take those. Do write them down. Email them to me. I am available on Slack. I'm also available on my email, and I'll be happy to answer them later on. Yeah? Awesome.
Yes.
Thank you very much. Nag, are you still there?
Yeah.
Okay, so I will leave these brilliant kids in your hand and I'll carry on. Is that fine with you?
Oh yeah, sure. Yeah, absolutely. So
Okay,
thank you very
guys,
much. Thanks
thank
for
you. No
putting
issue.
those kind pictures.
Awesome. Yeah, I
I
found
don't
them.
know, I thought like, you were closing it with this class or something. That's why you wanted to thank Lakshmi, Mittal and everybody.
No, this is just for, you guys are being nice, being organizing. So, yeah.
Good. So you can stick around. I'll wind it up quickly. Yeah.
Okay, I'll stick
Okay.
around. Then.
Yeah. So fantastic, guys, students, and sorry for those of you who still have some questions. I would say hold on to them, and this class is going to go for like next 4 sessions, right? So
Yeah.
make sure that you're not running out of questions and using all your questions in the first class, right? So that's also an important thing for you to keep it up. That's good. Yeah. As fun as all of this going on, I'm slightly worried about the assignment part.